So the last speaker for this session will be Professor Ehud Gazit from Tel Aviv University, who will give us a talk about amyloid fibrils in metabolic disorders and neurodegenerative diseases.
Thank you, Reut. Thank you, the organizers, for the opportunity to speak today. Thank you for getting my... Yes.
With them in one.
I need to
It's okay.
Yeah. I'm very punctual, so I need to... I'm stopping my stopwatch.
There it is.
Okay.
Up, up. Oops. Now it will work. רגע.
Does it work?
Yeah, it will. Now it's
Great, thank you very much. So today I would like to speak about amyloid. You heard about amyloids in this conference, but with a twist. I don't have a lot of time, but I would like... now I can see it. Okay. Amyloids are related to some of the most devastating age-related disorders, including Alzheimer's disease, Parkinson's disease, ALS, Huntington disease, as others. As many of you know, my personal background is more studying the molecular and biochemical properties of self-assembly of molecules, and amyloids were a prime example for us. Try to understand what is the exact mechanism in which proteins and peptides form well-ordered structures. Our approach over the years was reductionist, minimalistic, non-biased. What does it mean? We take complex biological molecules, we cut them into smaller and smaller pieces, and try to identify the shortest fragments that can form ordered assemblies. In the case of the beta amyloids relate to Alzheimer's disease, this is almost a 20-year-old paper. Initial knowledge was on a short fragment that can form amyloid fibers, two fragments that can inhibit the formation. The common denominator was just a dipeptide motif, diphenylalanine, and we demonstrated already 18 years ago that this very simple peptide can form well-ordered structures. Not classical amyloids, but that share many properties with classical amyloids. A few years later, modifying a little bit the termini of this dipeptide motif, we were able to show that just a simple dipeptide, acetylated in one hand and amidated in the other, can form ordered assemblies that look like classical amyloid that many of us are familiar with. So this was our reductionist amyloid scheme, starting from a 42 amino acid peptide, going to a hexapeptide, then a dipeptide. We also could see that the dipeptide structures can form physical entities of unique properties. For example, mechanical one. We could see that just the dipeptide can form structures that have the mechanical rigidity similar to bone or teeth. So it was in 2005. A few years later, it was shown by Thomas Knowles from Cambridge University and Marcus Bühler from MIT that amyloids that are involved in human disorders have similar mechanical properties. We could see other properties such as piezoelectricity, the ability to have electrical current upon the application of mechanical forces. Optical properties for the structure that's being formed. Again, later on, few years later, another group in Cambridge could demonstrate this same phenomena with amyloid structures taking from full-length protein that are involved in disease. And we could see other properties that are, this is my other side of my split personality, showing properties like semiconductivity of amyloids and other. The reason that I'm stressing this is, first of all, I think it's quite interesting, but more importantly for this audience, as just in the next few slides, I will show you the ability of even smaller elements to form ordered assemblies relates to biology, and as you will see, they have also very similar physical properties as were shown by us and others for very short peptide, as well as amyloids that are related to disease. So all I want to speak about in the 15 minutes left is relate, or even less, Reut is looking at a watch, is something that started as a control. We were studying the ability of diphenylalanine to form such ordered assembly. We studied other related peptides, including triphenylalanine, tetraphenylalanine, other short aromatic peptides. And until less than a decade ago, if someone would ask me what is the minimal requirement to form these assemblies that share properties of structures that are being formed by disease-related amyloid forming peptides and proteins, would be the one peptide bond, which due to its unique chemical properties have this partial two-dimensional properties and allow the stacking of the structures one on top of the other. So testing this hypothesis, we went on to study whether a single amino acid could form amyloid fibers. The answer should have been known. And we selected phenylalanine because phenylalanine is, as I said, it's was a component of many of the peptides that we studied. And quite to our surprise, we realized that phenylalanine, just by itself, but relatively high concentrations of millimolar and higher, could form structures that looked like amyloid on the electron microscope. Those of you who are working with amyloids, they bind Congo red and show the typical green apple birefringence between cross polarizers. They could bind THT. They showed ordered structure, ordered electron diffraction pattern, just like classical amyloids. Yet, the concentration of phenylalanine in which the amyloids were formed were completely non-physiological. The concentration of phenylalanine for each one of us, I assume here in the audience, even if we had a lot of proteins during the breaks, is somewhere between 40 to 60 micromolar, and I'm speaking about formation of amyloids in concentrations of millimolar. Yet, after scratching our heads, we realized that there are conditions in which you have phenylalanine concentration of millimolar. This is in diseases known as inborn of metabolism. In this case, it's phenylketonuria. Any one of you, children, you pinch the toe in when the child is born to measure the level of phenylalanine in the newborn. And indeed, for children affected with this disease, the concentration of phenylalanine is actually in the range of millimolar. The clinical definition of phenylketonuria is a phenylalanine concentration above 1.2 millimolar. And this is due to a recessive mutation in the enzyme that converts phenylalanine into tyrosine. And we later on realized that similar to amyloids that are related to age-related disorders, you see toxicity, which is concentration dependent. Again, seeing toxicity around a concentration that are related to the pathological concentration of phenylalanine with patients. In the past, it was assumed that toxic effect that are seen with a very high concentration of phenylalanine relates to some imbalance of transport of amino acid and other phenomena. Yet, we had a very nice control. We took alanine, which does not form such amyloid structures, and even at concentration of 15 millimolar, we could not see any toxicity. Moreover, we could raise antibodies toward this towards these fibrils, and by using these antibodies, could deplete the toxicity of the phenylalanine fibers. We could also see the spontaneous generation of antibodies towards these fibers in a mice model of PKU. Human and mice have exactly the same mechanism of formation of phenylalanine, of tyrosine, processing of phenylalanine, so you can delete this gene and get the accumulation of phenylalanine in the mice. We could even see, and this is becoming more and more related to what you see in old age, we could see deposits in brain of PKU patient, post-mortem of course, of these amyloid-like fibers co-stained with the antibodies as well as Congo red. And this is very similar to things that we see with age-related disorders. So we could see our reductionist amyloid scheme went down from a dipeptide to a single amino acid. Unfortunately, phenylalanine is not the only metabolite to be accumulated in what is known as inborn of metabolism, these diseases in which you have some enzymes that are related to biological pathways, and you have a list of these metabolites and many more that are being accumulated in various disorders. And again, in a very systematic way, we studied all the list. Some people now study additional metabolites, and we could see that not only phenylalanine, but also adenine, orotic acid, cysteine, tyrosine, uracil, now it's known the tryptophan, other metabolites can form things that look like amyloid. They bind THT like amyloid, they bind Congo red like amyloid, and moreover, they induce late apoptosis, just like amyloids that are made of beta amyloid or alpha-synuclein or other proteins that are related to age-related, are associated with age-related diseases. So in the mechanism of late apoptosis, and this is for the entire list, and you always have this control of alanine. You put, so for the non-scientific term, tons of alanine on the cells, and you see nothing. But with those structures that can form amyloid-like assemblies, you could see a clear behavior that is like in the case of amyloid disease. Now, I'm going back to the physical properties, again, reflecting what we are very much interested in those assemblies. So we could see, then not long time ago, that these structures that are made by the metabolite, either by adenine, tryptophan, tyrosine, phenylalanine, show the optical properties just similar to what we could see with the dipeptide motifs, and what could be observed with disease-related. So same properties with amyloid-like structures that are found to be formed by metabolites, found to be formed by peptides, and found to be formed by disease-related proteins. Same for the mechanics. So it's this is our work more than 15 years ago, the work that I mentioned later, and just a few months ago, we could demonstrate the same thing with the structures that are made by phenylalanine. this is true also for the piezoelectric property, seem to be not related whatsoever to biology, although we have some ideas about what happens when you have something that induces electrical current in your brain. So we think that it might be more physiological than previously appreciated. But also this, not by us, but by other group, was shown that just structures that are made by glycine amino acid are piezoelectric. And we have now yeast models for the formation of amyloid-like structures using the metabolites. We see behavior, again, that is very similar to what we see with the classical amyloids, including this very cooperative way of formation of amyloid structures and sensitivity of yeast models for the feeding with the metabolites. We could rescue the yeast by using, in the beginning, just classical polyphenols that are known to inhibit amyloid formation by proteins and polypeptides. Now we have unpublished results on also screening for more effective therapies. And an important part is that we can rescue the cell by the treatment without reducing the concentration of the metabolite. So in the past it was assumed that you have huge concentration of metabolites, and this is the reason for the disease. We suggest, and we have more and more evidence, that is probably that it's probably the formation of the structures. So to summarize, before a few more minutes about relation again to all the age-related disorders, is we see that not only proteins, polypeptides, dipeptides could form structures that have an ensemble of properties that are common, but also structures that are made by simple metabolites, amino acid, nucleobases, vitamins, small chemical entities that are accumulated in various diseases. Thinking about this, and we try to understand what may be the interplay between the formation of structures that look like amyloid by the metabolites and other diseases that are related to old age. So if we think about a disease like homocysteinuria, a cysteine can form amyloid-like structures. They have 20 times higher concentration of cysteine as compared to a normal individual. But some individuals with 20% higher concentration, 30% higher concentration, you see a very high tendency to form this disease. So we ask him, maybe the seeding of proteins by metabolite assemblies may clarify some unexplained epidemiological associations. And two connections between metabolites and amyloid formation by proteins. One is quinylonic acid. We know that quinylonic acid, high concentration of quinylonic acid are related to Parkinson's disease. And we could see that indeed quinylonic acid can form structures that look like typical amyloids, and we also solve their structure by crystallography and see a typical amyloid-like structure. And we could demonstrate the seeding of alpha-synuclein, which is not tend to form amyloid so easily by those seeds. And quite recently, we could see that in the case of homocysteine, which is a marker for Alzheimer's disease, we could see the formation of typical fibrils by homocysteine. We have a yeast model for this, I will skip it, but with the mice model, the 5X FAD that Uri Asheri was mentioned in his talk, one of the best models for Alzheimer's disease, we could see the colocalization of beta amyloid and homocysteine, which is well related to our hypothesis about the co-aggregation of metabolites and proteins. So I think that in the last 10 seconds, we think it's a common phenomena that you see with proteins, peptides, very short peptides, metabolite, and we think there's a crosstalk between those entities that should be further studied. So simplicity is the ultimate sophistication. It's not my saying, but Leonardo da Vinci, people around, and I guess that's not much time for questions.
So thank you, Professor Gazit, for the intriguing talk. We don't have time for questions, but people are welcome to ask Professor Gazit later.